Hello, this is Professor Torsten. You're watching the recorded lecture for Chapter 8, Chromosomes and Cell Division. This chapter is looking at cell division, how cells divide. We will be looking at the process of mitosis, which is how most of your body cells divide. We'll also be looking at the process of meiosis, which is how your body produces sperm if you're a man, or eggs if you're a woman. We'll also take a look at what can happen when there are errors or mistakes in cell division. We've looked at chromosomes in this class already. Chromosomes essentially are DNA. When we look at the difference between prokaryotic cells and eukaryotic cells, we see that there is a difference in the chromosomes. Prokaryotic cells, which you remember are the smaller, simpler cells like bacteria, have one circular chromosome, as you can see here in figure 8.3. Eukaryotic cells, which include animal and plant cells, have linear chromosomes. They usually have several linear chromosomes, and those chromosomes are stored within a nucleus. Because of this difference, not only in the chromosome structure, but the difference in the structure of the cells, that eukaryotic cells have this nucleus and prokaryotic cells do not, there is a difference in the way prokaryotic cells and eukaryotic cells divide. Prokaryotic cells divide through a process called binary fission. This process is shown in figure 8.4. Towards the top of the figure, we see one prokaryotic cell. We are calling this the parent cell. Within this parent cell, we see one circular chromosome. And remember, prokaryotic cells don't have a nucleus. So this chromosome is simply inside the cell membrane. The first step for cell division requires DNA replication. We want to make an exact copy of the chromosome so that each of the resulting offspring cells have a copy of DNA. So here in the middle of the figure, you see one prokaryotic cell with two circular chromosomes. Those two circular chromosomes are identical. We then see the process of the cell splitting and dividing in half to produce two daughter cells. The daughter cells are identical to each other. The daughter cells are also identical to the initial parent cell. This process of binary fission is an example of asexual reproduction. The process of eukaryotic cell division is more complicated than how prokaryotic cells divide. Eukaryotic cells store their DNA within a nucleus. Now, eukaryotic cells still have to replicate their DNA just like prokaryotic cells did. The parent cell produces a full copy of the DNA so that the resulting daughter cells have a copy of the cell's DNA. Eukaryotic cells can divide through mitosis or by meiosis, and we're going to be looking at these two processes in the rest of the chapter. Here in figure 8.6, we're looking at an overview of the eukaryotic cell cycle. We see that it looks like a circle, and arrows indicate that it moves in a clockwise manner. Most of this circle is taken up by this purple arrow, and this purple arrow is representing a stage of the cell cycle called interphase. Here in blue, we are looking at the mitotic phase. So M here stands for mitosis. Within this cell cycle, there are several checkpoints. and they are represented in this figure by this red and white striped bar. These are checkpoints. Checkpoints are critical points in the cell cycle in which progress is blocked. So cells are actually prevented from dividing until they receive specific signals indicating it's okay to continue the process. We will look at what happens when cells ignore these checkpoints, because cancer often results from cells ignoring these checkpoints. Here we look at interphase in eukaryotic cells. Figure 8.5 shows the eukaryotic cell cycle, and again, the textbook is using purple to indicate interphase. So you can see interphase is taking up most of the cell cycle. Interphase can be subdivided further into gap1, S phase, gap2, and gap0. If we look at gap1, this is where the cell spends most of its time. Gap1 is normal cell activity, normal cell function. Here we see S phase. S stands for synthesis. The cell is synthesizing DNA. Remember, the cell needs to replicate all of its DNA so that it can give a full set of DNA to each of the two daughter cells it will form. After S phase comes gap2. Gap2 is a period of high cell activity as the cell prepares to enter the cell division phase or the mitotic phase. Gap0 is a resting stage. The cell is not actively dividing in gap0. Some cells will stay in gap0 for days or even years, and some cells will never enter gap0. It really depends on the type of cell that you're talking about. Let's take a closer look at S phase. S stands for synthesis. During S phase, the cell is synthesizing DNA. Remember, the cell needs to copy all of the chromosomes, and it will end up with twice as much DNA as it usually has. So if we consider human cells normally have 46 chromosomes. But after completing S phase, human cells have a total of 92 chromosomes. Here in figure 8.7, we're looking at an overview of the DNA replication process. DNA replication is based on the same base pairing rules that you guys have learned already. Remember, A and T pair together, and G and C pair together. What that means is that if we start here on the left-hand side of the figure with the parental cell's DNA, if we separate these two strands, as shown here, we can use each of these two strands as a template. So if we separate this left strand, and we can see this left strand has T, A, G, C, T, A from top to bottom, the cell will synthesize the complementary strand simply by following those base pairing rules. So here it reads A on the parental strand, and it knows A pairs with T. Then it sees T, and again, it knows the base pairing rules and will synthesize A next, and so on. The same thing happens in the opposite strand. The result is that we have two copies of DNA where we started with one copy, and each of these copies is identical. They're identical to each other, and they are identical to the DNA that we started with. If we look a little bit closer at DNA synthesis, here we see in figure 8.9 what the chromosome is really looking like. So here we see the double helix, the spiral of the DNA, and we see that the two strands of DNA have been separated. Once these two strands of DNA are separated, these enzymes, called DNA polymerase, can synthesize the complementary strand. So here on the left-hand side in this darker turquoise is the parental DNA strand. DNA polymerase, here in orange, is creating a new strand by following those base pairing rules. The same thing is happening over here in the parental strand on the right-hand side. So every time a cell divides, it needs to copy all of its DNA. And remember, it needs to do this so that each of the two daughter cells it forms has all of the genetic material for the cell. Now, before we continue, I want to introduce a couple glossary terms. We've been talking about chromosomes, as represented here in figure 8.13. Once chromosomes have been replicated during S phase, they'll look something like this here in the middle. It almost looks like a letter X. Each one of these blue lines is a sister chromatid, and they're held together here in the center in a region called the centromere. These sister chromatids are identical. Now let's take a closer look at mitosis. We're looking at the same figure here, figure 8.5, and we've looked at interphase, which is this purple region. Mitosis is represented here in blue, and during mitosis, the cell is actively dividing. We see division of the nucleus first, and then division of the cell body itself. So before we get into the stages of mitosis, I want to talk a little bit about why cells need to undergo mitosis. So most of all your body cells, and the fancy name for body cells are somatic cells, divide using mitosis. Mitosis enables cells to generate new genetically identical cells. And this is how eukaryotic organisms grow bigger. Their cells divide, and there are more cells, and therefore the organism itself is bigger. It's also how eukaryotic organisms can replace dead, dying, or damaged cells. The rate of mitosis, how quickly cells divide, can vary greatly depending on the type of cell. So for example, red blood cells are replaced every 2 to 4 months. The lining of your intestines, those cells, are replaced about every 3 weeks. And things like bone cells are replaced every couple of years. So it really depends on the type of cell that you're talking about in terms of how quickly mitosis happens. So now we can look at the stages of mitosis as outlined here in figure 8.11. At the top of the figure, we see the parent cell. Here in purple is the nucleus, and inside the nucleus is all the DNA. So during interphase, the cell will copy or replicate all of its DNA in S phase. Once the cell enters mitosis, it will proceed through 4 stages called prophase, metaphase, anaphase, and telophase. The end result is the formation of 2 identical daughter cells. These daughter cells are identical to each other, and they are identical to the parent cell that we started with. Prophase is the first stage of mitosis. Figure 814 shows you an overview of what the cell would look like in interphase as compared to prophase. So if we look at the cell in interphase, we see the nucleus here in purple has a bunch of DNA. All of these almost looking like spaghetti noodles, these are all chromosomes. An image taken from a fluorescent microscope shows a real-life cell here, and in purple is the nucleus. In prophase, three main things happen. Number one, the sister chromatids condense. On the left-hand side in interphase, we see all of these chromosomes as really long, almost spaghetti noodle-like shapes. On the right-hand side in prophase, we see all of the sister chromatids have condensed or compacted down so that they almost look like the letter X. So these are sister chromatids held together at the centromere, which is this center dot. We can see this in the figure above. Here in purple, again, is the nucleus, and these really dark purple spots are the condensed sister chromatids. The second thing that happens in prophase is that the nuclear envelope, or the nuclear membrane, actually disappears. You can see this more easily in the drawing. Here is the nuclear membrane, and you can see it's starting to fade away. The third thing that happens in prophase is the formation of the mitotic spindle. The mitotic spindle is made up of a bunch of proteins, and these proteins are the actual cell machinery that causes the nucleus to divide. So here we see centrioles. There's one here and there's one up there. So there are two centrioles, and these centrioles are where those mitotic spindle fibers, those proteins, are growing out from. You can see those spindle fibers here in green in that fluorescent image at the top of figure 814. The second stage of mitosis is called metaphase. In metaphase, we still see our sister chromatids in their condensed or compact form. Again, they almost look like a letter X. And in metaphase, these chromosomes line up in the middle of the cell. We can see this in the fluorescent image above. All of these purple lines here are sister chromatids. These sister chromatids have lined up in the middle of the cell because these protein fibers in the mitotic spindle have latched on to the sister chromatids and physically pushed and pulled them until they are lined up in the center of the cell. Notice that in metaphase, there is no nuclear envelope or nuclear membrane because we got rid of it during prophase. So during metaphase, the cell actually does not have a nucleus. The third stage of mitosis is called anaphase. During anaphase, the sister chromatids are pulled apart. So remember in the previous slide, we saw that sister chromatids looked like the letter X. Here in anaphase, we see that the letter X has been broken in half so that one sister chromatid ends up on one side of the cell and the other sister chromatid ends up on the opposite side of the cell. So anaphase is pulling those two sister chromatids apart from one another. Again, the thing that is actually physically moving the sister chromatids are the proteins of the mitotic spindle. These are represented by these light blue lines here in figure 814. The fluorescent image above shows you, again in purple, the chromosomes being separated towards opposite ends. In anaphase, we still don't have a nucleus because we still do not have that nuclear envelope or nuclear membrane. The fourth stage of mitosis is called telophase. During telophase, the cell reforms the nucleus by reforming the nuclear membrane or nuclear envelope. Here in the figure, we see a drawing of a cell in telophase, and we see all the chromosomes here on opposite ends of the cell. And in purple, we see that nuclear envelope beginning to reform. Telophase is a really interesting stage because it's the only time we ever see a cell with two nuclei at the same time. So one cell has two nuclei, and each nucleus has a complete set of DNA. We can see this in the figure above as well. Finally, the cell will undergo cytokinesis, which is division of the cell body itself. In telophase, we saw the nuclei reform at opposite ends of the cell. And in cytokinesis, we see division of the cell body itself so that we get two separate daughter cells. These two daughter cells are identical to one another, and they are identical to the parent cell we started out with. A few slides ago, I mentioned that cancer often results when cells ignore those checkpoints in the cell cycle. Cancer itself is unrestrained or unregulated cell growth. So oftentimes, cells will ignore those cell cycle checkpoints and continue dividing even when they're not supposed to be. Every time a cell divides, we just learned the cell has to copy all of the DNA. And this, of course, leaves the opportunity for mutations to arise. In Unit 2, we learned that mutations are simply changes in the nucleotide sequence. So for example, instead of GAC, there's a mutation and we end up with GCC. Now our cells do have proofreading and repair enzymes that look for these mutations and fix them, but our proofreading enzymes aren't perfect, so some mutations may not get corrected. In terms of cancer, there are three features of cancer cells that make them different from normal cells. Normal cells have something called contact inhibition. Normal cells will divide until they touch other cells. And once they sense that there are other cells around them, they stop dividing. Here in figure 815, we see a petri dish filled with cells, and there's one layer of cells here. Once these cells sensed the edge of the petri dish, as well as all these neighboring cells, they stopped dividing. Here on the right-hand side, we see cancer cells are ignoring that contact inhibition. Cancer cells ignore the fact that there are plenty of cells in this petri dish, and they continue dividing. So you can see there's almost a pile of cancer cells here in this petri dish. So cancer cells can lose their contact inhibition and therefore continue dividing even though there are more than enough cells present here. A second feature common to cancer cells is that cancer cells can divide indefinitely. So most of our cells have a limit of how many times they can divide. So most of our cells can divide 80 to 90 times, and then they'll stop. The cell might continue living, but it won't divide any further. Cancer cells, though, seem to never lose their ability to divide. So they continue dividing, and that is part of the reason why we have unregulated cell growth in cancer. A third feature of cancer cells is their reduced stickiness. Normal cells, as pictured here in 815, these normal cells pictured in blue are being held together by proteins in their cell membranes. For more about proteins in cell membranes, I refer you back to Unit 1. Cancer cells have reduced stickiness or adhesiveness, which means they don't stick together as well. This means that cancer cells can easily break away from where they started and move to other regions of the body, something called metastasis when cancer spreads throughout the body. A couple slides ago, I mentioned that most cells have a limited number of times that they can divide. Most cells are able to divide 80 to 90 times and then they stop. Part of this can be linked back to telomeres. Telomeres are sections of your chromosome that are actually not coding for anything in particular. So they're not specific genes, it's just a protective bit of DNA. So here in figure 8.1 in blue is the functional DNA or the coding DNA. So here in blue are actual genes, and these genes of course could be for just about anything: hair color, eye color, etc. At the end of this chromosome in green, we see a region of the chromosome called the telomere. And again, the telomere is non-coding, so there are no genes in this green area representing the telomere. Every time a cell divides, it misses the very end of the chromosome. So you can see after this first round of cell division, this last little bit of the chromosome is not copied. On the next round of cell division, again, we lose that last little bit of the chromosome. If this continues, eventually we would be losing important DNA here in blue. So these telomeres act as protective end caps so that every time the cell divides and loses the ends of the chromosome, it's only losing the telomere region and not the coding or important region of the chromosome. So telomeres can help keep track of the number of times a cell divides, and when the telomeres get too short, the cell does not divide further. Some cells have the ability to rebuild their telomeres. So some cells have an extra enzyme that when the telomere gets too short, it can extend it back longer. So far, we've talked about mitosis, which is how somatic cells divide. Somatic cells are all the cells in your body except for your gametes. Gametes are sperm for men and eggs for women. Now, most of our cells are diploid. We have 2 copies of every chromosome in our cells. Gametes, though, are interesting because gametes are actually haploid. Sperm and eggs have half the number of chromosomes than the rest of our cells do. This means that when a sperm and an egg come together, these 2 haploid cells combine to form a diploid cell. Figure 8.18 on the right-hand side of the slide is showing you an overview of this process. Humans normally have 46 chromosomes in total because we have 2 sets of 23 chromosomes. So sperm and eggs each have 23 chromosomes, so that when they combine and fertilization happens, we end up with an organism that has the correct number of chromosomes, a total of 46 chromosomes. Meiosis has some steps that are very similar to mitosis, but it also has a couple important differences. Meiosis is necessary for sexual reproduction because it reduces the amount of genetic material in gametes. Remember, humans are diploid organisms, so our cells normally have 46 chromosomes in total, 2 copies of each chromosome. Gametes, our sperm and eggs, however, are haploid. They only have 23 chromosomes each. This means that during fertilization, 23 chromosomes in sperm combine with the 23 chromosomes in the egg to create a cell that has 46 chromosomes in total. The advantage to sexual reproduction is that it increases the genetic diversity in a population. Meiosis is actually 2 rounds of cell divisions. If we look at figure 8.21, we see we are starting with a diploid parent cell. Diploid meaning 2 copies of every chromosome. Now in this figure, we're simplifying it, so we're only looking at 1 pair of chromosomes here. The darker green color is the maternal chromosome, the chromosome you inherited from your mother, and in yellow is the paternal chromosome, the chromosome you inherited from your father. These 2 chromosomes are called homologous chromosomes, or the homologs, or some people will call them the homologous pair. Homologous chromosomes are not identical. Again, they are 1 maternal and 1 paternal copy of the chromosome. Now, these chromosomes have the same genes. For example, this green chromosome from mom might have the gene for eye color, and this chromosome from dad might have the gene for eye color, but the actual instructions might be different. So for example, the eye color gene on the maternal chromosome might have the set of instructions for blue eyes, and the gene here on the paternal chromosome might have the set of instructions for brown eyes. When DNA is replicated during S phase of interphase, we know that we are copying this DNA so that we end up with 2 identical copies of that chromosome that we called sister chromatids. And these sister chromatids, remember, are held together at their centromere. So we see 1 set of sister chromatids of the maternal chromosome in dark green, and we have 2 sister chromatids here in yellow coming from the paternal chromosome. So when we go back and we look at figure 8.21, we look at the parent cell, and we start out with 1 green and 1 yellow chromosome. During interphase, specifically during S phase, these chromosomes are replicated. So we end up with 1 set of sister chromatids for the green chromosome and one set of sister chromatids for the yellow chromosome. In the first round of meiosis, we see these chromosomes separate. And then in the second round of meiosis, we see these sister chromatids separating so that we get the formation of 4 haploid daughter cells. And these are the gametes, the sperm or the eggs. Here in anaphase 1, we see that homologous chromosomes are separating, but the sister chromatids are staying together. So if we look at these blue chromosomes here in the center, we see that these 2 sister chromatids are still together, and these 2 sister chromatids are still together. The second round of cell division in meiosis is when the sister chromatids separate. Again, we still have 4 stages, but we've called them prophase 2, metaphase 2, anaphase 2, and telophase 2 to indicate that we are talking about the second meiotic division. Again, the key steps are very similar to mitosis. In prophase, we see chromosomes condense down, we see the nuclear membrane break down, and we see formation of the mitotic spindle. Here in anaphase, we see the separation of sister chromatids. And by the end of meiosis, we will have formed a total of 4 daughter cells, and those daughter cells are haploid. They have half the amount of DNA as the parent cell did. The advantage of sexual reproduction is that it increases the genetic diversity of a population. The increase in genetic diversity comes from 3 main factors. Number 1, we are combining the genetic information of 2 people. Number 2, there is something called crossing over. And number 3, there is something called reassortment of homologs. Crossing over is when chromosomes will actually swap portions of each other. So here in this darker blue color, we see a set of sister chromatids from mom. And these 2 dark blue chromosomes are identical to one another. They are sister chromatids held together at the centromere. And in light blue, we see another set of sister chromatids coming from dad. And again, sister chromatids are identical. Now these 2 darker blue sister chromatids and the 2 lighter blue sister chromatids are not identical to one another. These are homologous chromosomes. You can see that these 2 chromosomes have crossed over one another, hence the name crossing over, and they've actually traded portions of each other. So you see this dark blue sister chromatid has part of the light blue chromosome, and this light blue chromosome has part of the dark blue chromosome. The third way to increase genetic diversity is through reassortment of homologous chromosomes. What this essentially means is that the way the chromosomes separate from one another during meiosis is random, so that we don't get all of the maternal chromosomes in one gamete and all of the paternal chromosomes in another gamete. There's a mixture of chromosomes. One error that can occur in meiosis is something called non-disjunction. Non-disjunction is when chromosomes do not separate correctly. So in figure 835, we're looking at an overview of meiosis. Here at the top of the figure, we're looking at metaphase 1. what we see are two light blue sister chromatids attached at the centromere and two dark blue sister chromatids attached at their centromere. The two light blue sister chromatids and the two dark blue sister chromatids are homologous chromosomes. The same holds true for these green sister chromatids as well. So these dark green sister chromatids are identical to one another. The light green sister chromatids are identical to one another. And all of these together are the homologous chromosomes. So during anaphase 1, normally we see homologous chromosomes separate. Now the blue chromosomes did this correctly. We see the dark blue sister chromatids went to the right-hand side. The light blue sister chromatids went to the left-hand side. So the blue homologous chromosomes separated correctly. However, the green sister chromatids did not separate correctly. We can see the dark green sister chromatids and the light green sister chromatids all ended up here on the left-hand side of the cell. When the cell goes through the second round of meiosis, or meiosis 2, we see that there are issues or errors in the resulting gametes. On the right-hand side, we see gametes containing one chromosome. So each of these gametes only have one blue chromosome. They are missing a green chromosome. These are gametes, so these are either sperm or eggs. And what that means is when this gamete from this parent combines with a gamete from another person, the result is going to be an individual that is missing a chromosome. On the left-hand side, we see these gametes have 3 chromosomes each. These gametes have an extra chromosome. They should have 1 blue and 1 green, but they have 1 blue and 2 green, again, because the green homologous chromosomes failed to separate correctly during anaphase 1. Each of these gametes then have an extra chromosome. So when this gamete combines with the gamete from another person, the resulting offspring is going to have 1 extra chromosome. There are a number of human conditions that arise from this. The most common is Down syndrome. Down syndrome is when a person has 3 copies of a particular chromosome. We can look at a karyotype, which is a visual display of a person's chromosomes, and we can see here we have 2 copies of chromosome 1. So one of these is a maternal copy, the other is the paternal copy. Here is chromosome 2. Again, one of these chromosomes is the maternal copy and the other is the paternal copy, and so on. If we look at the bottom row, this is chromosome 21, and there are 3 copies of chromosome 21. So this is an extra chromosome. That concludes the recorded lecture for Chapter 8.